Good afternoon. The title of my talk is Genetic Factors Associated with Multiple Sclerosis Risk and Clinical Characteristics in the Kuwaiti Population. I am the project's principal investigator, and my co-investigators were Dr. Raed Al-Roghani, an MS consultant from Amir Hospital, and Professor Fahad Al-Mulla from the Faculty of Medicine, Kuwait University. I'm going to start off with an overview about multiple sclerosis. So what is multiple sclerosis? It's a chronic inflammatory autoimmune neurodegenerative disease of the central nervous system. The pathogenesis of MS is believed to be infiltrating immune cells, mostly T cells, that are autoreactive to myelin and myelin-associated antigens that cross the brain-blood barrier, invading brain's parenchyma, causing axonal demyelination and oligodendrocyte death, cell death. Across time, repeated MS attacks or inflammatory episodes result in demyelinated plaques, which we refer to as MS plaques on MRI, throughout the central nervous system, causing irreversible perturbed sensory and motor nerve conduction. There are 4 types of MS classified according to the rate of disability increase across time. The most common form is relapsing remitting MS, which is RRMS. The second most common form is SPMS, which is actually the natural course of RRMS. It converts into SPMS following an average of 12 to 15 years. The rarest form of MS is primary progressive MS. It is more severe, accumulates disability at a faster rate, and a more conspicuous, less clear form is primary relapsing MS, which is kind of going out of the classification of MS types. The global burden of MS appears to associate with the North Pole, hence the theory that vitamin D deficiency is a risk factor for MS. It has been shown to be so, including in our cohort. However, the prevalence of multiple sclerosis in Kuwait, which is a fairly sunny country, appears to be going upward. There's a trend of an increase, as can be seen here. In the past 2 decades, the prevalence of MS in Kuwait has increased from 4.4 MS patient per every 100,000 Kuwaiti individual up to 85 MS patient per every 100,000 individual. And this has forgot this part. The main MS characteristics that we know is that there is sex bias. The sex ratio is approximately 2 females to 1 male in terms of incidence. And in certain MS population, that ratio has shifted to a higher ratio of 3.2 to 1. The most established MS risk factor is the HLA-DRB11501 risk allele. We have assessed this risk allele in our population, and we found that it's not associated with MS risk in the Kuwaiti cohort. In addition, this risk allele is believed to explain only 20 to 60% of MS genetics. In total, there are so many studies out there about genes associated with MS risk, and I've included here the total number of those with replicate studies. There's approximately 107 genes involved with MS risk, and they encompass several functional groups such as motor proteins, cytokines, T-cell receptors, protein modification enzymes, single transducers, and many, many more. So what I'm presenting to you today is results from a part of a whole project. Our initiative was to actually assess all known or thought to be risk factors associated with MS. And we've already completed the environmental factors assessment, infectious agents, circulating biomarkers, and what I'm presenting today is the genetic profile of the MS cohort, primarily the exome sequencing results. Our experimental pipeline involved recruitment and collection of MS patients and healthy controls that are purely Kuwaitis. We had our own criteria of inclusion, and we tried our best to approximate sex and age matching. Samples were processed, exome sequencing was performed on Illumina HiSeq 2000 at a 50x coverage. Sequence QC was performed using online programs, and analysis was done using 2genomics application. The main objectives of this study were the following: is to identify genetic risk factors associated with MS as a whole. Second, to identify genetic factors associated with familial MS risk. And then third is we were hoping to identify genetic predictors that associate with MS clinical course as well as response to treatment. So this table shows the study cohorts. We had 113 MS patients and 80 healthy controls. They were all Kuwaitis. We managed to approximate sex ratios. We also managed to approximate the age. We were matching them. Now, in terms of MS type, as I mentioned, our RMS is the most common form, hence it comprised the bulk of our MS cohort. Second to it was the SPMS and PPMS. In addition to that, a positive family history of MS is believed to be a risk factor for MS incidence. And as you can see here, we had 50 MS patients that reported a positive family history of MS up to the 5th degree of relation. EDSS score is a disability score. It stands for Expanded Disability Status Scale. And as you can see, again, the bulk were within the range of 1 to 2.5, which is expected because these patients are not disabled up to a degree that they will not be willing to provide a blood sample. They had more initiative to participate in the project. But lucky for us, we also managed to get quite a good number in both of the other groups on the EDSS score. So before we started off looking for novel Kuwaiti MS genetic risk factors, we consulted the International Multiple Sclerosis Genetics Consortium, or IMGC. They performed multiple studies, genome-wide studies of multiple MS populations, specifically focusing on European Caucasians. There are 53 genes reported, excluding those on the X and Y chromosome. Unfortunately, we found out that the majority of these variations in these 53 genes were actually non-coding, but we consulted our exome results looking for the coding SNPs, and we found contradictory findings. Whereas they found these two first variants to be risk factors, they appear to be protective in our population. So we decided that our population is most likely distinct from European Caucasians, so we moved on to look for novel Kuwaiti MS genetic risk factors. And as expected, HLA loci were there, and they were there at high significance rates. As can be seen here, you have HLA DRB1, DRB5, A, B, in addition to DQA1, DQB1. But we had several novel genes involved, such as CFAP221, and based on gene ontology, it really is promising for us. PLIN4 also is promising, so is LGAL-S9B. We haven't replicated these findings yet. We hope to replicate them in the future. We are performing HLA allelotyping at this point. The next objective was to determine familial MS genetic risk factors. So in a case control setting, we had 15 MS patients with a positive history of MS and 78 healthy control Kuwaitis. We replicated the association of the HLA loci and other genes, SNPs, that we found in the previous table that I showed you. However, most interestingly, an olfactory receptor gene appeared to associate strongly with familial MS risk. So we went research hunting to see whether anybody reported something similar to this. And we found in a single study, there was an association of mate selection among MS families. This study studied 14 MS families that had an established familial MS within them, and they found that mate selection, the parents themselves, was based on similarities in HLA and olfactory receptor variations in their MS dataset. In addition, olfactory dysfunction appears to be common in all MS types, And an MS autopsy cohort, this is a separate study, reported that the most MS-related demyelination actually occurs in parts of the olfactory pathway in 71% of post-mortem MS cases, post-mortem autopsies MS cases. So what did we find? We found the same as I just mentioned. Olfactory receptor had the highest p-value, followed by the same HLA-DRB1 SNP and the same CFAP221 SNP, in addition to the olfactory receptor, the secondary SNP down below there. Again, we are in the process of confirming these results. Our third objective, we were just basically hoping, and we got several findings. So we wanted to investigate why do male MS patients progress more severely and faster than female MS patients. They accumulate disability at a faster rate than female MS patients. So to investigate that, we tested 8 MS males versus 19 female MS patients' exome sequences, and these two, they were less than the original cohort because we compared their disease duration, but they had variable disease progression. And based on the results, we assessed based on gene ontology and possible variant effects, a SNP in plexin A3 or PLXN-A3, which is a gene found on the X chromosome, was the most significant finding. What is PLXN-A3? It is a protein that is required for normal dendrite spine morphology in pyramidal neurons and is important for cytoskeletal remodeling following injury and axon pathfinding in the nervous system. So in order to confirm our findings, we had a replicate cohort of 184 MS patients. We genotyped them for the SNP and PLXN-A3, and we found that allele G associated with higher MSSS. MSSS stands for Multiple Sclerosis Severity Score, so it measures severity across time, and increased EDSS, the disability scale, when compared to allele C male MS patients. In addition, allele G was also found to increase the risk of an aggressive MS clinical course, specifically in male MS patients. And as you can see here, due to the fact that the EDSS number, the majority of our cases had low disability, the relative risk was substantial, but the confidence interval was approaching 1, but nevertheless, it was still significant. Here I'm just highlighting to you where the mutation is. It is actually part of a structural domain that is very important to the function of PLXN-A3. The second finding is that we found a pharmacogenetic predictor of response to natalizumab. Now, natalizumab, or you might know it as Tysabri, is actually a second-line treatment for multiple sclerosis, very effective, but about 30% of the patients do not respond well to Tysabri. They have adverse effects, or they actually, the disease progresses with them. There's no halting of the disease progression course. And we found that a mutation in GP6, which is a platelet protein, appears to associate with thrombocytopenia and poor response to natalizumab. I will not go into the details because my student, Malak Al-Mu'jiz, poster is tomorrow. It is poster number 20. If you need further details, kindly go see her. So to conclude, the Kuwaiti population genetic predisposition to MS appears to be distinct from other populations, sharing minor similarities, and I'm here referring to the HLA. Familial MS risk is influenced by olfactory receptor and HLA loci variations, possibly by parental mate selection. Male MS severity is influenced by genetics, referring to the PLXN-A3 mutation. And the concluding remark is that exome sequencing of multifactorial disorders can be productive, given that the cohort is representative of the disease both quantitatively and qualitatively. Before I end, I would like to acknowledge that this work was kindly funded by Kuwait Foundation for the Advancement of Science, KFAS, and special thanks go to my students, These are all med students, undergrads, and best of gratitude goes to my technician, Sindhu Jacob. And thank you for your attention.